Okay, we are live now.
To the old, long life and treasure. To the young, all health and pleasure. Well said by Ben Jonson. I think the best is yet to come. I welcome you all for this wonderful evening. 1 week FDP program on statistical analysis, academics, and research. Before we start our second day, I would like to mention here a special thing that we are having international speakers to motivate us for our FDP, which is going to be a really good thought. So today on board, we have Suchitra Bhattacharya ji, research scholar, Stevens Institute of Technology, Hoboken, NJ, USA. I welcome you, ma'am, on board. And before we proceed, I would like to make some announcements for the participants that all the updates related to the FDP is available on our official website, www.theijms.com. Related to the FDP, whatever queries and your questions, you can visit to the website for your updates. Now, I welcome Suchitra Bhattacharya ma'am to start her presentation. Over to you, ma'am. Suchitra ma'am, are you hearing me?
Kindly unmute, ma'am.
Am I audible?
Yes, ma'am, you are audible.
Yes.
Okay. So good evening, everyone. I am in different time zone. So I welcome you all to the India's biggest FDP organized by IJMS. So first, as Ma'am Alia told that we are here for a on the talk for like on statistics, how why statistics is important and what are its importance in the research study. So first, I would like to say what is statistics. It's a branch of mathematics which deals with collection, handling, and processing of data, interpreting the results in a very logical manner. Statistics is very useful in every discipline, and we use it in our day-to-day life. I would like to point out some of the functions. Like first comes the predictions. Predictions are made on the basis of data which we collect and something which might happen in near future. Like doctors used to use this prediction tool to know the fate of the disease. Then recently, John Hopkins research published a COVID-19 model, which helps to determine if the patient's condition is getting likely to be worsened. Then comes the census. It is a complete evaluation of each and every individual, which makes the population. It is one of the important statistical tools used by both government and non-government organization, I assume in every country, to know the characteristic of the whole population. Last but not the least is the sampling. So sampling is the portion of population which is to be analyzed, and it is very easier to understand the characteristic of the whole population. In certain situation, when a government or non-government are you everyone is can you hear me?
Yes, ma'am. Yes.
Okay.
Yes, ma'am. You are audible, ma'am.
Okay. So when it is not possible to collect the whole census, the government and non-government organization do sampling to know the characteristic of whole population. So it is mostly used in medical science to know the prevalence of any disease. Now I would like to go a little in deep about why statistics is specifically important in our research study. So it is very paramount because it contains description, inference, and communication. The results which we get in our study are much more objective, and interpretations are done through statistical model. So here I would like to say a little about statistical software which I used during my master's is a GraphPad Prism. This is advanced software, and it is commercial scientific 2D graphing and statistics software. Following are some of the features which I'm going to say, like it provides statistical guidance for the novice. Then there are many nonlinear regressions with many options. Live links, if you want to edit or replace, Prism automatically updates the results and graphs. Analysis choice can be reviewed and changed at any point of time. Then there are automatic error bars too. Now I would like to point some of the features of statistical analysis, why it is important in research study. So first, it informs method on data collection. At first, one need to identify which test is required for the particular research study. Then statistics helps to identify which you do not consider while collecting data. Then it used to support or negate a hypothesis. If there is no statistical tool, then there is no way whether the data supports the research or do not. Moreover, if we didn't use statistical tool or there is no statistics, then we would have buried in many unknown questions and open-ended conclusions. Then it also helps to avoid uncertainty, errors, and outliers in the data. So we are human, we do errors, but which is unavoidable. But if statistics is not there, then there is no way to avoid such errors. Statistics help to understand the level of uncertainty in our results and help us to deal with it very fairly. Although no data set or statistical tool is perfect, but at least we can draw conclusion with the help of statistical tool by understanding the limitations from the very beginning. Now, last but not the least is it helps aid interpretation, summarization, and communication of data sets. With the help of statistics, we are able to interpret the data set accurately and can make an imperial decision what the data actually means. And the best way is to communicate the result and help us to defend why we chose this particular statistical tool or statistical software and how we came to this conclusion. So I hope you find it helpful. And thank you, IJMS, especially IJMS, for giving me this opportunity. So thank you and have a nice day.
Thank you, ma'am. It was really short and sweet, and it was wonderful to know many good things about statistics. So now, moving to our session of the day. And I would like to start this with a quote here. Never underestimate the power you have to take your life in a new direction, said by German Kent. So today we have a session 1 on analysis of variance or ANOVA. And today's speaker is Dr. Sunita Panikar. Dr. Sunita Panikar is Assistant Professor, Department of Microbiology, Dr. D.Y. Patil, Arts, Commerce and Science College, Pimpri, Pune. Now, I would like Meenal Majati to give the profile of the speaker. Over to you, Meenal.
Thanks, Nazma. Good evening, everyone. Sometimes reaching out and taking someone's hand is the beginning of journey. And such a beautiful journey of togetherness we all are experiencing in one way. Faculty Development Program arranged by International Journal of Microbial Science in collaboration with Sushila Shankarrao Gadhave, Mahavidyalaya, Khandala District, Satara. It's my honor to be the part of FDP. And here I'm myself, Meena Masti, here to introduce our today's first speaker, Dr. Sunita Panikar ma'am. Ma'am has completed her PhD in biotechnology through CSIR, JRF, and SRF fellowship, and then was awarded ICMR postdoctoral fellowship at National Institute of Epidemiology, Chennai. Before her PhD, she has worked as a research assistant at NARI Pune and as a senior research fellow at Hafkian Research Institute, Mumbai. Currently, she is working as an assistant professor in Department of Microbiology at Dr. D.Y. Patil Arts, Commerce and Science College, Pimpri, Pune. She has many peer-reviewed research articles to her credit, few of which are published in Elsevier and Springer journals, and has also authored 2 textbooks for the undergraduate microbiology students. Last year, Dr. Panikam ma'am was awarded ICMR International Travel Grant to present her work in a HBV meeting at Melbourne, Australia. Dr. Sunita ma'am is a member for 1000 Girls, 1000 Futures, as she is a member of Organization of Women in Science and for Developing World, Strada Costeria, Italy. And this is really great to hear, ma'am. She is on the editorial board of few prestigious journals and reviewed many manuscripts. She is actively involved in research related to virology and molecular biology. This is a brief introduction of ma'am, and now I request Dr. Sunita Panikar ma'am to take over the session. Please, ma'am. Thank you.
Thank you for this sweet introduction. Thank you, ma'am. Good evening to all the participants. Am I audible?
Yes, ma'am, you're audible.
Yes. So, I'm here to talk on ANOVA as the ma'am has told in the introduction. So, these are few of my articles published in Elsevier and Springer journals. And so, before starting the lecture or the talk, I would like to make a humble request to all the participants to have a pen and paper with them, so that in the next few slides, when we are actually going to perform analysis of variance manually, I want you to do it with me. So please have a pen and paper with me. Okay, so let's begin. What is ANOVA? We all know what is ANOVA. For brief, at least we know. ANOVA is a commonly used or most widely used technique which is used in comparing the measurement of data. It is like when you have you perform an experiment or you do some survey or you collect a data, you want to analyze it, you want to interpret what experiment you have carried out, okay, and that analysis you perform on that data is your ANOVA, okay. So mean, comparing the mean. So ANOVA, we all know that it is an abbreviation for analysis of variance. It was invented by R.A. Fisher in 1918, and that's why it is also called as Fisher analysis of variance. Okay. So if we want to officially define ANOVA, what is that? Analysis of variance is statistical technique that is used to check if means of two or more groups are significantly different from each other. That means we are analyzing the means. Okay. So ANOVA checks what? It checks whether the impact of one factor in your experiment, okay, in your experiment, whether one factor is different or it is more better than the other factor, and that if you want to check, that is if you want to find out whether there is difference in the test groups or the control groups, you do the ANOVA. Okay. So when we say ANOVA, invariably we talk about variance. So what is variance then? We need to define that also. Variance, the average square difference from the mean, or variance measures the spread out of your data. It measures how variable is your data. Right. So after talking so much of 2 slides on variance, analysis of variance, then we said means, all of this might be a bit confusing, like actually what it is, whether we are analyzing the variance or we are analyzing mean. So what are we doing actually in ANOVA, whether it is mean or variance? Okay, so let me explain that to you. Actually, when we apply statistics to a data, right, when you perform an experiment and you want to interpret, what you do statistics. Means what? You draw conclusions or you interpret your results, right? Now interpret your results, and from what? From the population parameters, or you just check which one is better by doing the mean, taking out the mean, right? So in both of these cases, what is misleading? Misleading factor is your mean. When you say only simple mean, then what happens? It is just comparing the two means, right, to find out which one is better. But when you do this, at what level you can say that this mean is better than the other mean, or mean one is better than the mean to. When you want to do this at a confidence or a higher confidence level, and you want to say that for a long period of time, this will be the status. That mean one will be always better than the mean to. When you want to say that, what you want to do, then you want to analyze the variance, and that is where your variance comes into picture. Okay, so more the variance, your lower is the confidence that your mean one will be always better, or is for a long term, it is always better than the mean two. So this, when you want to say this with a high level of confidence, your variance should be less. And that is the reason what we do. We do the analysis of variance, or you analyze the variance by studying the mean. Means of what? Means of the response variables. That is the factors, levels that you perform in your experiment, right? So to determine the confidence level, to make a judgment based on the mean, what you do is you analyze the variance. So now I think this explanation might have made a bit clear concept of what is analysis of variance. It is actually the analysis of mean through analysis of variance, right? So you are analyzing the mean by using the variance. So the name, that is ANOVA, is appropriate because inferences about the mean are made by analyzing the variance. Okay, so I think it is a bit clear concept that I have made. I've tried to make. Okay, so now when you talk about ANOVA, the special feature of ANOVA is that you have two things to analyze. You just don't look only in the difference in the group 1 and group 2, but what you do, you have a concept called as within the group and between the group, right? So what is that concept? When you say that you have three groups, okay, when you say you have three groups, you just don't look at the variability between these three groups, but what you look as well, you look at the variability within the group also. Okay, so ANOVA has a special feature, or to say that, or to look or to analyze between the group and within the group. Okay, so the variability within each group is a measure of spread of the data within the group. Okay, in the same group, if it is group 1, then what is the variability of each reading from the mean. And then when you talk about the between the groups, that is group 1, group 2 and group 3, then the variability between the groups is the measure of spread of the group means. Okay, so group 1 means how it is spread from the overall, the grand mean. Okay, so that is you have to first consider this concept very clearly, that ANOVA has a variability checking between the groups and within the group. Okay, so you say that your ANOVA is the average variability between groups divided by average variability within the group. Okay, so F statistics, what we said, it is called as Fisher statistics also, or Fisher's ANOVA also. So F statistics, it is also called as F statistics. Now in F statistics or ANOVA, what we have is we have two assumptions. Okay, we need to make two assumptions. And now what are those assumptions? Or you can call them as hypothesis also, or more tendency is to call them as hypothesis. So first hypothesis is null hypothesis, and other one, so in null means what? Null is zero or none or nothing, right? So what nothing or no, what we are analyzing? We are analyzing mean. And what we are looking at, difference of the mean. So when you talk about null hypothesis, what it would be? There is no difference in the means of all the groups. Okay, so that is all about your null hypothesis. And exactly opposite to your null hypothesis will be alternative hypothesis. Alternative to null. Null opposite to your null will be your alternative hypothesis. So alternative hypothesis, what is your null hypothesis? That all the means are equal. Okay, there is no difference. That is your null hypothesis. So opposite to that would be there is a difference, or one mean is at least better or different from all the means. So that will be your alternative hypothesis. And why you want to do these assumptions, or why you want to make the hypothesis is depending on these hypotheses, you get the when you do the calculation. Okay, when you do the performance ANOVA then what you get the results out of this, when you want to interpret those results, that will tell you whether to accept the null hypothesis or to reject the null hypothesis. When you accept the null hypothesis, that will be that all the means are equal and your experiment is not very giving you great results. And when you reject the null hypothesis, what it will be, that your experiment is showing some difference in the means of the group. So you can go ahead and find out, right? So that is the reason you have to make the hypothesis. It's a major thing in the ANOVA. Now ANOVA is classified into two ways. That is one-way ANOVA, which is also called as a single classification ANOVA, and a two-way ANOVA. Now let's see what is one-way ANOVA. Now one-way ANOVA is considered when only one factor is used. A simple experiment where you have a set of data depending on only one variable or one factor. Okay, so to determine whether the means of two or more independent groups are statistically significant using one factor, just one factor. So I will explain this with an example. Let's take the performance of students based on the anxiety levels. Anxiety levels means what? We can group them into low, medium, and high stress students. Means what? The anxiety level we have grouped into low, medium, and high stress. And what we are looking at? We are looking at their exam performance. So what we have, this is a very simple experiment. We are looking at the low, medium, and high stress students and looking at their exam performance. So what? We have only one factor into consideration, and that is your dependent variable, that is your factor. What factor it is? It is the anxiety level, the stress level of the student, right? So in this case, you will be using ANOVA, which is one-way analysis or one-way ANOVA. Okay, so remember, when one factor is into consideration, you will use one-way ANOVA. Now two-way ANOVA, when the you find the difference between the means that have split into two different independent variables. Now, what does that mean? If we extend the same previous example, like the student's performance at the based on the anxiety level. Now, this anxiety level is one factor that we have considered, but if you further go and split this anxiety level into gender and education. Gender level means gender male and female, whether the male show different anxiety level, female show different anxiety level. Education level means education, whether they are PG students who are less stressed, or the UG levels are less stressed. What's that? Okay, so what we have done, we have split out the dependent variable into two groups, into two independent variables. So in that case, what you will do, you will use a two-way ANOVA. Okay, so I hope it is clear when to use one-way ANOVA and when to use a two-way ANOVA. So gender and education will be your independent variables, and the dependent variable will be the anxiety level. Okay, so clear where to use a one-way ANOVA and where to use a two-way ANOVA. Okay, so one-way ANOVA, we take into account only one variable, and in a two-way classification, you take two variables into account. And the primary purpose, the main purpose for doing a two-way ANOVA is to find out whether there is an interaction between the variables and the independent variables and the dependent. Like, for example, in our example, what we can say that whether the education PG level students are more stressed or not. Okay, so that is the interaction. Or whether the male are more stressed than the females, or females are less stressed than the male. So this is the interaction between the stress levels or the anxiety and the independent variable. So I hope it is clear that when to use a one-way ANOVA and when to use a two-way ANOVA. Okay. So before we solve this or we apply ANOVA to a data, let me take you through some terminologies that we will be using in ANOVA. First is a grand mean. We all know what is mean. That is the arithmetic average, right? So when you talk, it is the total of the readings upon the number of readings. So when you say grand mean, it is the irrespective of the group or the samples. You take everything into consideration and then you divide it by the whole number. So grand mean is the mean of all the numbers. Okay, all the samples and their numbers. So total of all the numbers and their samples. Then you have something called as degree of freedom. It is the freedom to vary between and vary within and within the total. So I told you that in ANOVA, always you have to consider every analysis between and within, between and within. So nothing can be out of this. So you cannot exclusively say that this is only for between, this is only for within. Everything you will be doing it for within and between the group. So degree of freedom also you will be doing it for within group and between group. And here are some formulas we'll be looking into it, right? Then you have something called as F critical. F critical, to find out the F critical, we use the F distribution table, and that F distribution table are in the statistical books, and how to use them, it depends on the degree of freedom between and degree of freedom within. And in the example, I will be showing you how to use this table. Then we have next is sum of squares. Sum of squares is the how each variation observation varies from its mean, right? So again here we have sum of square within and sum of square between. So variability within the group and between the group. Then we have mean square within and mean square between. That is your average variability. Now next we will be calculating your average variability, for which you will be requiring your sum of squares and the degree of freedom. Okay. And then lastly you have the F statistics. Finally, using all this calculations, you will calculate the degree of, you will calculate your F statistics, that is your mean square between divided by mean square within. And now there are 5 very easy steps to do this. Okay, to do one-way ANOVA. First, if you know how to do a one-way ANOVA, it's very easy to apply the same rules to your two-way ANOVA. Now first is your make the hypothesis and set the p-value. Hypothesis, we learned what it is. Your null hypothesis, where you will have all the means are equal, and then alternative, the means are not equal. P-value, this is the thing that we say that in biology, we always set the p-value at 5%, that is 0.05. Significant level we set at 0.05% or 5% or 0.05. The p-value will be 0.05. Then you calculate your degree of freedom within, total and between, and from that you will be calculating your F critical, that is your second step. Next step is to calculate the sum of squares within, between, and then from where you will get the sum of squares, for that you will be calculating your group means and your grand means. Okay. Next is to calculate the variance, that is your mean square. Again, you will be using your sum of squares and degree of freedom for that. And lastly, you can calculate the F statistics using the formula between upon within, right? And then after doing these 5 steps, final step is to compare that what you have got. What you have got, you have finally got the F statistics and you have got the F critical from your all these calculations. Now from these calculations, when you compare your F statistics with your F critical values, you can conclude whether your null hypothesis has to be accepted or rejected. If it is accepted, means what? There is no difference. If it is rejected, means what? There is difference, right? So I think these all 5 steps, if you follow them, it is very easy to solve or you perform analysis of variance on a data. So let's take data, small data I have taken for an example, and please use your pen and paper. I'll dictate you the values and you can solve this manually. I'll be showing you how to manually solve or do analysis of variance on the small data. Okay, so let's start group 1, 1, 2, 5, 2 and 3. Then group 2, 2, 4, 2, 1 and 2. Group 3, 2, 3, 4, 2 and 4. Okay, so this is your data, and as you can see that the groups are not split anymore further into independent groups. As we talked in our earlier slide, what analysis we'll be using? It will be obviously one-way analysis. Okay, so one-way ANOVA will be using. So first step what would be? It will be stating the hypothesis. And what will be the hypothesis? First is null hypothesis, all the means. Mu is your mean. So mu1, mu2, mu3, that is group 1 mean, group 2 mean, and group 3 mean. They are equal. There is no difference. And alternative hypothesis, what would be that? That there is a difference and they are not equal. Okay. And then what I told you, set the p-value. So significance level, alpha level, we have set it at 0.05. Next up, what we said, we will be calculating the degree of freedom, the between, within, and total. Now, this degree of freedom between is the formula is k minus 1. Now, what is this k? The number of groups. How many groups do we have here? We have group 1, group 2, and group 3. So, 3 groups. So, k is equal to 3. So, 3 minus 1, it is 2. Okay, so your degree of freedom between is 2. The next is degree of freedom within. So that is the formula is n minus k. Now what is n? N is the total number of all the samples that you have or all the readings that you have. Now here each group is having 5. So 5 3s are 15. So your n will be 15, right? So how many k? We know what is k. It is the number of groups. So how many? 3. So it is 12. So degree of freedom within is 12. Now, simply total will be addition of your between and within. So, your degree of freedom total will be 14. Okay. Now, next step is to find out the F critical value. Now, this F critical value, we can find it out from the F distribution table. Now, what is that and how to do that? This is your F distribution table at 5% level. We said that we'll be using your 5% significant level. Now, this horizontal lines, they are between, degree of freedom between, and these vertical ones, they are the degree of freedom within. Okay, this is available, this table is available in the statistical book. So, when you have this table, then this all are your between, these all are your DF within. Now, we have calculated between DF also and we have calculated within DF also. Between how much we have got? We have got it as 2. And within how much we have got? We have got it as 12. If you remember, if you are doing with me, right. So, what you have to do? You have to just find the point of intersection of these two degree of freedoms. And what is that? It is 3.885. Okay. So, you have understood how to use the table, the F distribution table. This is called as F distribution table. So, we have found your F critic value in this way. Now, next step here, that is you are finding the sum of squares. For that, what will require? We will require the mean values. Now, what are the mean values for the first? We will be finding out the mean values for all the individual groups. Now, here we have group 1, group 2, group 3. So, you have to find out mean for this group, and this group. So, what is the mean? Addition of all this divided by 5. So, what I did? 1 plus 2 plus 5 plus 3 plus 2, that is 13, and divided by number, that is n, that is 5. So I get it as 2.6. Okay. So next group is 2 plus 4 plus 2 plus 1 plus 2. So total I am getting 11. So 11 by 5, I am getting my group 2 mean as 2.2. Group 3. Okay, you are doing with me, right? So 2 plus 3 plus 4 plus 2 plus 4. So 15, and here you get 15 by 3. So you get your group 3 mean as 3. Right? Now we have to find out the grand mean. This X double bar is your grand mean. Now grand mean, what we said, irrespective of the group's total number. So total number is 15. So your n is 15. And then total of all this will be 13 plus 11 plus 15. Right? We did it the total here. So that your grand mean comes to 2.6. Okay, you got it. How we got the grand mean also now. Now we have to find out the sum of squares. Sum of squares, first we look at the total. Now this is your summation, that is your sum. Now what we have to do, we have to find out each observation or each reading, how it differs from the grand mean. Okay, variability. So how it is, how much it is differing. So this is your grand mean. That's so we are for total you have to remember that you will be using grand mean. For within you will be using your within means every group. So each group. So Here I'm using my grand mean, or grand mean is how much? 2.6. Okay, so this much part, please pay attention and do it very carefully and understand that what I'm using. Here I'm using my grand mean because it is the total, the sum of square total. So we will be using grand mean. So 1 minus 2.6 square plus 2 minus 2.6 square because your grand mean is 2.6. So same way 5 minus 2 minus 3 minus, okay, and square plus this ends my group 1. Now plus same thing I'll be doing it for my group 2. So group 2 first reading 2, so 2 minus 2.6, that is my grand mean. 4 minus 2.6, 2 minus 2.6, 1 minus 2.6 and 2 minus 2.6 square. Everybody is square. Then I do it again plus. Same thing I'll be doing it for the third group. So third group first reading is 2. So 2 minus 2.6, 3 minus 2.6 plus 4 minus 2.6, 2 minus 2.6 plus 4 minus 2.6 again. Right? So there ends all the three groups. So S total, the SS total or the sum of square total will be 19.6. Okay, if you do all these calculations, you can do this, verify these calculations after the lecture. But for now, take this as the value. So you have your sum of total as 19.6. Now let's solve your sum of square within. Sum of square within, you will be doing the additions for all the three groups. Okay. So every reading of each group minus the mean of that group. Okay. So this x1 is the mean of group 1. x2 is the bar x2. Okay. Bar x2 is the group of mean of group 2. Then bar x3 is the mean of group 3. So what I did, 1 minus 2.6, because 2.6 here, unfortunately the mean of group 1 is also 2.6. So don't confuse between your grand mean and the mean of your group 1. Okay, so please pay attention that this 2.6 is the x1. This is your x1 here, 2.6. Okay. So 2.6, 2 minus 2.6, 5 minus 2.6, 2 minus 2.6 and 3 minus 2.6 all squared. Plus, now this ends my group 1. Now plus 2 minus 2.6. Now I started with my group 2. So group 2 mean is how much? That is x bar 2. That is 2.2. So here I'll be taking 2.2. So 2 minus 2.2, 4 minus 2.2, then 2 minus 2.2, then 1 minus 2.2 and 2 minus 2.2. That ends my group 2. Okay, plus 2 minus 3. Now 3 is my group 3 mean. So 2 minus 3 square plus 3 minus 3 square plus 4 minus 3 square, 2 minus 3 square and 4 minus 3 square. Okay, so doing all these calculations, how much you get your S within? SS within is 18. Okay, so total, if you want to do, you have got the total, you have got the within, and when you want to find out the between, you just subtract your within from the total, and 19.6 minus 18, you get it as 1.6. Okay, so I hope you are doing all this with me. Good. Now next fourth step is to calculate your mean square. Now mean square, you have a formula that sum of square between divided by degree of freedom between. So we have calculated both, right? If you remember degree of freedom, how much it is? 2, and sum of square between 1.6. So 1.6 divided by 2, it is 0.8. Then same for within, SS within and DF within. So 18, we calculated it, your 18 as your sum of square within divided by DF, that we calculated previously in your earlier slide 12. So my answer comes to 1.5. Okay, and the last step, what we said, we want to find out the F statistics by using the formula MSW, that is your MSB, that is mean square between divided by mean square within. Okay, so how much we have calculated? 0.8 divided by 1.5. So the value comes to 0.53. Okay, now what we have? You have your F critic value from the table, if you remember. That is how much? 3.88. Now we have what we have to do. We have, why we did all this? We want these two main values, this and this. F statistic value and F critic value. For these two values, we did all these calculations. Remember this very carefully, that for these two values, we did all these calculations. Now from this, what you have to interpret? You have to look which one is greater. Here in this example, which one is greater? Your F critic is greater. Your F statistic is less than your F critic. And what does that tell you? That tell you that null hypothesis cannot be rejected, or you fail to reject your null hypothesis. And what was our null hypothesis? Means what? In other words, what we can say, we have to accept your null hypothesis. What is that? That there is no difference in the means of all the groups. Okay, so the experiment that you have performed showed no significant result. That will be the conclusion of this data what we analyze. Okay, so are you getting what I'm trying to explain you? Now the same thing, okay, the same analysis that we did manually, just look at this table. This is an Excel sheet, and you can do the same thing by just one click in your Excel. Okay, you have to just take your table into Excel, import it, or you can just type it again here in the Excel sheet, and then what? You have something called as a data. Click here, data tab here. Then if you click that data tab, what happens? Okay, there is something called as data analysis over here. Now this data analysis, if you are using the Excel software for the first time for statistical analysis, I'm telling you, not for any other thing, but if you are doing it for the first time for statistics, then maybe this data analysis tab is not there in your Excel, and you need to bring it to the Excel. And for that, how to bring your data analysis, if you are interested, you can put it into the chat box, and we'll be posting a video for that thing, like how to bring your data analysis tab. Okay, sometimes you will not find this data analysis over here. So next thing is what? When you click at this, what is this data analysis? It is a tool of all the statistics. It has all the tools in it, right? So what it will give you? It will give you, when you click at that data analysis, it will give you a pop-out, and this will have all your tools for statistics. That is your ANOVA, then correlation, regression, and all this, F-test, T-test, all the tests. Okay, so what we have decided, and the data is appropriate for doing a one-way analysis. So here is your ANOVA single factor. So you just take OK to your ANOVA. Okay, so single factor, because you want to perform a single one-way ANOVA. Now what happens? This window pops out. Now what is this? It is, I have asked the Excel to perform a ANOVA single factor on my data. What is this? It is what will it ask for? It will ask for you to put the input of the data that you want to analyze. Now how can you put that? It is this red color button here, the input range it is asking. This input range will be the numbers. Okay, when you have to take into consideration only the numbers and not the names. Like this group 1, group 2 are my names, and you will not be using this. When you click on this, you are selecting the input range. So when you click on this, you can just select all these numbers that you want to analyze, not the name. Okay, so this dotted line that is seen on the screen is the selection range that I have selected. Okay, now another important thing is, now my data is grouped according to the columns. Okay, and it is grouped by the columns and not by the rows. If I write my groups like this, then I'll have to select this another option, that is your rows. Okay, normally what we do this way only. The data is grouped by columns, but in case you have not done this and you have done it the other way around, that is you have grouped it by rows, then mind that you have to select your rows very carefully. Okay, you have to click this button. Now next is your alpha level, the alpha value that you want to do. by default it will tell you that the 0.05 or 5% level it has selected, which is okay, and leave it like that. The next option is output options. Output options mean what it is asking you, you can where you want the output, the table of analysis that will be generated, where you want that table to be. Either you want it in the same, that is the output range, means you want it in the same sheet, to select a cell and ask it to put it in that cell. Or there is another option of new worksheet. You can ask it to put it into another worksheet, or there is another option of entirely a new workbook. So doing all this, what you will do, just click OK. Okay. So when you do all this, you have to just click OK, and then what happens when doing this, you will get this table generated. Okay. Now this table is what we did manually only. See, what we calculated manually, we calculated the average, we calculated the sum of squares, we calculated the degree of freedom, we calculated the mean squares, then we calculated the F statistic value, then we had set our P value, and we calculated the F critic value. Okay, so in one or two clicks, the Excel has done all the calculations that we did. Okay, so you have to appreciate the work Excel is doing for you, but I will appreciate that if you know to do all this manually, then only you will realize what are these figures, and you will be able to interpret the data that is given to you by the Excel sheet. Okay, otherwise you will be surprised. How will I interpret my data looking at all these figures? Okay, if you don't know what is F critics, you don't know what is F statistics, what are these values, then it will be difficult for you to analyze. Even if the Excel is giving you this sheet, it will be not possible for you to analyze and interpret your data. Okay, so there is one more example I would like to tell you here, because the example that we had taken showed our F value or F statistic value less than the F critic value. Now here is an example where of single way ANOVA only, that is one way ANOVA, where your F value is more than the F critic value. And what will that tell you? What would be your interpretation? Your null hypothesis will be rejected. And what does that mean? That there is a difference, or one of the mean or of the samples is statistically, or it is different. Okay, significantly different from the different populations. Okay, so this is just an example to show that this data is what you expect to get from the result. Like most of the times what we want our F value to be more than your F critic value. Okay, now after understanding all these calculations and about everything about most of the things about ANOVA, I want you to see that before going into, you know now what data should be analyzed by one way ANOVA, what data should be analyzed by two way ANOVA. But before that also, I want you to tell that when you think of doing ANOVA or applying ANOVA test for a data set, then you have to make few assumptions, and you want to see that your data fits into these assumptions, and then only you will get the valid result for your ANOVA. Otherwise, if your data doesn't fit into these assumptions, then you cannot apply, or the results that you get won't be valid. Now, what are those? Your dependent variables should be measurable, or it should be measured at the interval levels or ratio levels. That is, they should be continuous. Like for example, time in hours, or weight in kgs, or height in centimeters. Continuous they should be. Then your independent variable should consist uh, more the three or more independent groups. Okay, so otherwise you will be, if it is less, then you will go for t test and not your ANOVA. Then you should have a independence of observations. Now what does this mean? If your dependent variable is split into independent variables, like for example, you are analyzing some some diabetic tablets on some diabetic patients. So you want to see, you have split it into diabetic patients and then diabetic patients with cholesterol also. So you cannot use one patient who can be here also in group 1 also and in group 2 also. Okay, so that is what does this mean? then there should be no significant outliers. Now, what is significant outlier means? Um, See, in everybody's life, I think we have done, most of the microbiologists or biological people, they have done standard graph for something or other. For example, glucose. If we do a standard graph for glucose, we have ODs and we have the concentrations. And as the concentration increases, your OD also increases, and finally it is gradually increasing. But due to manual errors or due to some technical errors, sometimes bubble is introduced, sometimes the tube is not washed properly, or some error, okay, some error I'm saying, which is unintended, and it happens in your, and then what happens to the reading? The reading goes beyond, like it is not in the range, it goes either very far away or it goes very below. Okay, so those readings are called as outliers, and you don't want such readings in your data, and then you cannot apply your ANOVA to it. Then your dependent variable should be normally distributed. The normally distributed means the more frequent data should be near the mean, and far from the mean should be less frequent data. Okay, so normal distribution. And then lastly, it should be the homogeneity of variances. The variance should be homogeneous. That is, a different treatment should not change your variability of the observation. Okay, so what are the talking about so much of ANOVA? Let's come to the limitations. What are the limitations? Limitations means, what we can say is ANOVA is an omnibus test. Omnibus is like general test. Okay, you can call it a general test because in ANOVA you cannot perfectly say that this group 1 mean is better than the group 2 mean. You can just say that there is a difference, or all the means are equal or they are not equal, but which one is better you cannot say. And when you want to do that, you have to do some sort of test, okay, like least significant test, Fisher least significant, and there are many more. Okay, so that is the biggest limitation of ANOVA. Now, since I'm a biologist and more specifically molecular biologist, I would will talk something about statistics in biology. So, for example, I've taken a Drosophila melanogaster, the fruit flies. They have the glycogen content of the fruit fly was measured in one experiment. And what was the basis of the experiment? That the researchers found that the enzyme, that is phosphoglucomutase, which is responsible for glycogen synthesis also and breakdown. Okay, and then what they saw that the enzyme sequence, the sequence of this enzyme was variable in different flies, and this variation led to combinations of amino acid sequences in these flies. Okay, so what was the variation? So at site 52, this protein sequence had valine or alanine. Okay, alanine and valine, they are the amino acids. And then at 84 site, there was either a valine or a leucine. So taking into all these combinations, you get 4 combinations of amino acid in the slice. So you will have slice with VV, you will have slice with VL and AV and AL. Okay. So what the experiment was, whether this variation will affect the glycogen content of the slice or not. Okay. So do the amino acid polymorphism in the locus, that is the enzyme locus, will affect the glycogen content or not? That is your biological question. That is the biological experiment we have set into. So remember that biology, when you do an experiment in biology, the biological question is always about process. Whether X process will affect this, or whether a Y will affect this, or does X cause Y, and so on. So its biological process or biological experiments will be related, and their interpretations also will be related to that. So when you talk about biological null hypothesis, what will be that different amino acid sequences will not affect the glycogen content, or different amino acid sequences will affect, like your statistics. But when you look at the same experiment from the statistical point of view, okay, now you're coming to the statistical importance of statistics in biology. When you look at the same experiment from statistical point of view, what you will say, statistical point of view will be numbered. You will be looking at the number, the mean of the average of the glycogen content. If this combination of amino acid is there, what is the glycogen content? If this is there, then what is the content? So remember, biological process or experiment will be related to the process, and statistical will be related to the numbers. Okay, so it will be about the process, and statistical hypothesis always about the numbers. Your glycogen content in the numbers are either same or different. That is your statistical hypothesis. But if you talk in biological terms, the biology of amino acid sequences in the enzymes will be the amino acid sequence, the biological process. Okay. So now since here only one factor is used, that is your glycogen content, what ANOVA will you use? Will use a one-way ANOVA. Okay, so I hope that is clear with you. Importance of the statistics in biology, and then coming to the last point, that is the applications. Applications may, it is similar to t-test, but it is widely used, versatile test, and it is more in common, and you can use it in any field like pharmacy, biology, microbiology, agriculture, statistics, marketing, business, research, then finance, mechanical calculation, everywhere you can apply your ANOVA. Okay, and for example, like you can calculate the plant of height of the plants with or without galls. Then you can calculate the weight of the deers in different geographical regions. Then you have now the current COVID situation. So many people are trying for their vaccines and drugs. So what analysis they'll be putting into? They can use ANOVA for the statistical analysis. They're comparing the attention spans, right? The anxiety levels and all those things. Okay. So thank you for listening me patiently, and I hope I have added to your knowledge. Not very much, but at least a few points I could. It will be great if I have made some points clear to you. And if any questions, I'll be happy to take them. So thank you once again.
Yes, ma'am. Indeed, it was really a very good session, and we had a lot of clarity on ANOVA. Now, I would welcome all the questions from the participants. So, the question answer session is taken by Dr. Sheetal Banerjee, and I would request her to continue with the question answer session. Ma'am, you can take over.
Thank you, ma'am. All the participants are requested to post their questions in the chat box. Beginning the question answer session with the famous quote of Thomas Berger, the art and science of asking question is the source of all knowledge. I, Sheetal Banerjee, welcome you all in the question answer session of this FDP. First question posted by our esteemed participants in the chat box is, what is the meaning of single factor ANOVA? What are the assumptions of ANOVA and how to check this?
Uh, I have answered these questions in my presentation, but still I would like to answer. Single factor means when one variable is into consideration, like very simple experiment where you are analyzing only one factor. Okay, it is not split further into any groups or independent variables. And the assumptions are, there are 6 assumptions that you want to fit your data into, and that I have explained in the this. I don't think we'll get so much time to explain again the all assumptions here. So is it fine
Yes,
if I?
ma'am. Yes, ma'am. Thank you, ma'am. Thank you. One of the question which has been just now come in the chat box is that, can you please explain comparative analysis method? Comparative analysis method, ma'am.
Comparative analysis method, it will take a long time to explain that. This I think we have a session on that. So let's keep it for that session.
Okay, ma'am. Okay. Any other question if the participants want to ask, please you can put in the chat box so that we can keep it in front of our esteemed speaker for their expertise. Yes, ma'am. One question is that if F value is greater than F critic, but P value is greater than 0.05, what would be our null hypothesis?
Null hypothesis is always the same. We have to state the null hypothesis. means are not equal. Okay, then depending on that, you have to make your assumptions whether you want to accept the null hypothesis or you want to reject. Depending on the F critic value, whether your F critic value is less or if your F critic value is more, you will be accepting your null hypothesis or rejecting your null hypothesis. Now, talking about the p value, when you set your p value at point not 5 or the 5%, that means you are giving the 5% chance of getting what you say the level of confidence, or you can set say it as a cutoff value. So when you go more below than 5%, then you make it more stringent. Okay, so that will depend on your whether to accept the null hypothesis or to reject will depend whether it is F value is more, critic is more, or F value statistic is more.
Okay, thank you, ma'am. One last question is that if the same experiment is conducted in 2 consecutive years, then how data should be analyzed? Like
2 consecutive years means you have the data for same.
Ma'am,
for two
actually,
years? Yeah.
I would like to say that I did this one experiment in 2015 and another
Okay,
in the 2016, the same experiment with the effect of bacteria on nodulation,
okay,
say on nodulation parameter. Then
okay.
how I will analyze that, how I will write in the paper, like it's like the same. I have a 2 years data.
Yes, but in biology, you have the conditions. You cannot say that your 15 conditions will be same as your year 16 conditions or the next year conditions. So you will have to analyze the two data separately, and then you have to take into consideration the two analysis conditions, and then you will interpret your result. So you will be performing analysis of or ANOVA for your first year data and then your second year data, and look at the differences what you have done. Or see, biological process will never be same if you even if you so that is what your reproducibility of results is. Even if you say that, that is why we take the p level as the 5%. Okay, so because we have the degree of freedom to vary so much, so you cannot analyze same data set from that year and this year. You have to just compare them by doing the ANOVA separately for both the years.
And this is for the field data. It's like I have a field data and other is a lab data. So do you think that in the biological system we can the condition of 2015 and 2016 will be same
Yes,
in the
that's
lab
what.
data?
Now, see, yes, even see the media that you are using, the water that you are using, all these conditions will matter for your results. Okay, so you cannot compare or you cannot take into consideration the data. That is what a continuous it should be. So you can do it only on separate analysis on your year one data and year two data.
Understood, madam. Thank you.
Yes,
Thank you, ma'am. Thanks a lot for your expertise. On
thank you
behalf
so
of the
much.
organizing committee and on my own behalf, I owe my gratitude for your esteemed presence and your expertise as well.
Yes,
Thank you, ma'am.
I also
Over to you,
would like.
Alia, ma'am.
I would also
Okay,
like to
ma'am. Thank you, ma'am.
thank the organizers for giving me this opportunity. Thank you so much.
Our pleasure, ma'am. Thank you. Over to you, Alia, ma'am. Alia, ma'am, kindly unmute yourselves.
Huh. Every time I forget this. Anyways, now I would like to make some announcements before we proceed with the next session. So the first announcement is for the participants. The participants are hereby informed and updated on the thing that whatever you are having YouTube videos which are live streaming will be only until live streaming. There will be no recorded version on the YouTube. So I request all the participants to be present on live sessions and mark your attendance. That is very much important to get your certificates. The second thing is whatever updates you are requesting or wanting will be updated on our website, that is www.theijms.com. Next is we have a very good announcement here. And this is really interesting because we have a kind of test at the end of the 7 days FDP session on 8th November 2020. We have the test of MCQs consisting of 50 questions, to which 20% of marks is compulsory to receive your certificates. And the link for the same will be able on 8th November, 7 p.m. sharp, and it will be disabled by 8 p.m. sharp. So 7 to 8, the link will be open for you to answer the MCQ session. Out of this, there will be 3 best participants who will be getting their national level awards for the best participants, and it will be delivered to their address by post. Next important feature is the feedback. Everything we do, we require a good feedback to analyze where we are, what we have to do next. So with this thing in our mind, the feedback of the speakers and the link for the same is being provided on the website. So I request all the participants to give your feedback of the speakers, because again, we are having 3 cheers to the organizing committee here. The best 3 speakers will be rated by the participants, who will be then awarded national level awards for the best 3 speakers. So this is a great moment for all of us. So I think we should gear up to be the best and to receive the best. So the mementos which will be given will be delivered to the address by post. So this is the thing. And one more important notification I want to make here is, on the websites, we have already displayed the links of previous days and today's links of presentation and the PPTs. So whoever is interested can go through this again. And mind you all, time is everything and punctuality is the most important thing. So our FDP is 1 week FDP, online FDP. So self-learning is the mantra here. And so you all, after having your FDP, you can go through this. After that, the FDP links will be disabled on the 7th of November 2020. I remind you, 7th of November 2020, after we finish our sessions, all the links will be disabled. So make sure you are doing your updates regularly so that you don't miss out any of the important topics which may be very helpful for you in your career growth. With this thought, now let us proceed to our next session, and that is development of new and innovative courses and curricula, which is also the need of the hour. And we all have shifted from offline mode to online mode of learning and teaching. And this is one such kind of requirement now. So we will be gaining more insight. The speaker of the day is Kishore Kumar Dash, academic counselor, journalism and mass communication, Odisha State Open University, Malkangiri, Odisha. I request Meenal Majati to give the introduction of the speaker. Over to you, Meenal.
Thanks, Nazma. Once again, a very good evening to one and all. I'm again with you all to introduce the speaker of our next session, Kishore Kumar Das sir. Sir has completed Masters of Journalism and Mass Communication and currently holding the post like Academic Counselor, Journalism and Mass Communication, Odisha State Open University, Model Degree College, Study Center, Malkangiri, Odisha, and a teaching faculty with Department of Botany, Balimala College of Science and Technology, Malkangiri, Odisha. Some of the research papers published by Sir with title Decoding Magic Bullet Theory in Times of COVID-19 Pandemic has been published in UGC Care List Group 1 Journal Shod Sanchar Bulletin. Paper titled Rebooting Tourism in Post-COVID Economy has been published in Jabalpur West International Journal of Social Science and Management Studies. Some of his under-process research papers include Understanding Environmental Changes During COVID-19 Pandemic by Osmania University and Hyderabad Institute of Social Science. Addition to that, about at least 100 of articles been put into the print in various national level magazines like Geopolitics, Geography and Uday India. Kishore Kumar Das sir has awarded by Odisha Chief Minister Naveen Patnaik in 2012 for a story on the abduction of Sukma collector Alex Paul Manan. He has successfully participated and delivered talks on various conferences organized by UNICEF at New Delhi, Patna, and Bhubaneswar. Including this, he is members of Jabalpur-based International Social Science and Management Welfare Association. Now, I request Kishore Kumar Das sir to continue with the session. Please, sir. Thank you.
Hello. Hello, ma'am.
Yes, sir.
Am I audible?
Yes, sir. You are audible, sir.
I am also visible. Hello, ma'am. Am I visible and audible?
Yes, sir. You are visible and audible as well. You can start, sir.
Okay, fine. Thank you. So let me share my screen.
Yeah.
Here, participants of today's evening. So here you can see that you can see on my screen
Sir, your
that
screen is
what
not
a
visible.
chaiwala is doing.
Excuse me, sir. Your screen is not shared, sir. It's not visible. Now it's visible, sir.
Okay.
Yes,
Is
sir.
it
Now
okay?
you can tell.
So, dear participants, you can see on screen that what a chaiwala is doing. Simply, he is splitting hot tea from one jug to a glass. Now, why he doing so? Behind, and why the seller is doing this? So, please try to answer these questions. So, guessing the answer, keep finding the answer till I share my personal experience and a particular story with you. In a fine evening of January, month of January, I traveled my best friend. So I started my journey, in fact, from my hometown here in Odisha. For Bhubaneswar, the capital of Odisha. So after 100 kilometers, the bus stopped at a place.
Excuse me, sir. Your
As
voice
it was okay.
not very clear, sir. The voice is not clear, sir. It's not coming clearly. It's coming like very slow.
Is it breaking, ma'am? Is it breaking?
Yes. Yeah, it's breaking, sir.
Okay. Right now it is okay.
No, sir. Still, it's not clear.
Maybe some technical glitches, I think so.
Yes, sir. You may.
Should I remove earphones?
No, sir.
Okay.
Yeah, now you speak, sir. Let me check.
Okay. So, while on the way to Bhubaneswar, the bus stopped at a place, and all the passengers got down, and we also, myself and my friend, so and we went to a nearby tea stall for a cup. Ma'am, right now, is it audible?
No, sir. It's
It's still
audible, but
breaking, ma'am.
there is some issue, sir. We are not able to understand clearly.
Okay. What to do? Just a minute, ma'am. Just a minute.
Ashish sir, can you help it out?
Sure.
Ashish
Ma'am,
sir?
actually, it's a network problem.
Yes.
Yeah, I can't help.
Okay.
Okay,
Maybe sir, you can rejoin or reconnect your system to the internet. You can try this. You
let me
just
try. Okay, let me try from my side, sir.
yes, sir. I will suggest rejoin, and maybe you can reset your internet connection, then try to join.
Yeah. Okay, let me try.
I request participants to stay tuned. I apologize for the technical glitches happening. We will be right back. And meantime, I would like to make an announcement here. All the participants are requested to update yourself with the kind of going on. And today with our second session, um, I request all the participants to stay tuned too, because we'll be any minute be back. And apologies. Uh,
Hello. Hello, ma'am.
yes, sir.
Am I audible now?
Yes, sir. You are audible, sir.
And whether the voice is breaking or it is all right?
No, sir. Voice is clear, sir. You can share your screen and let me see how it works.
Okay,
Proceed,
fine,
sir.
fine.
Yeah.
Okay, just a minute. Okay, ma'am.
Yes, sir.
So right now, voice is clear?
Yeah.
Okay. Then should I proceed, ma'am?
You can.
Hello?
Sir. Yes, sir. You can proceed, sir.
Okay, okay, fine. So what I was telling, so when the bus stopped at a place, all the passengers got down and we also got down and went to a nearby tea stall near the bus stand. And we asked the chaiwala to prepare 2 cups of tea. Then what he told, yes, sir, you have to wait for that because whatever the tea I had, it has already been exhausted. So I have to prepare. I have to make fresh tea for you, but that you have to wait for another 5 minutes. We told, okay. So meanwhile, he prepared the tea for us, but it was too hot. Meanwhile, the bus driver was in a hurry and he was blowing the horn. And we were not in a position to take the tea as it was quite hot. Then what we decided to pay money of the tea and go. But the tea seller told us, no, sir, the tea is ready. Now you take tea. Just wait for 1 minute. So then what he did, brought one dog. Uh, hello.
Yes, sir. You are audible, sir. Please continue. Yeah.
Okay. Okay. My screen is visible.
Yes, sir. Yes.
Okay. Then the tea seller told us, sir, the tea is ready. Just wait for 1 minute. So what he did, he brought one, or you can say jug, and poured the tea to a glass. And within minutes, tea came poured, and we took our tea and got into the bus and the bus left for the destination. Now you may ask me, sir, why you are telling this story? In what way this story is related to your topic which we are going to deliver right now? So here, yes, my dear participants, I told you this story because I want to tell you that how storytelling is going to benefit students or the learners. That is number one. And number two, as I was telling you before telling the story to you, that what is the rationale behind it, what is the general rationale, and what is the scientific rationale behind it, and why the teacher is doing so. Yes, you know that had the job been there, the whole tea would have been cold. That is number one. So I told you this story because the capacity of an academic counselor, one day I was taking a class in my university, and university
Sir, you are not audible. Can you please check your speaker? Dash sir, kindly unmute yourselves.
Okay, ma'am, you please set the screen. Then I will start. Okay, ma'am. Hello. Hello,
Yes,
ma'am.
yes, sir, you are audible, sir.
Okay. Should I start?
Yes, sir, but still your voice is not clear, sir. It is breaking.
Okay, ma'am. Okay, let me try. It happens due to some technical issue, actually, which is beyond my control.
Yes, but it is now clear.
Okay. So what happened? So what is the rationale behind it, and why the chaiwala is doing so? The simple reason that he, with the help of that jug, he wanted to cool the hot tea before serving to us, and that is why he did. So while taking a class on the importance of parliament, particularly the Rajya Sabha, in my university in Odisha, so I was telling this story to them, and here the jug can be compared with the Rajya Sabha, and the glass can be compared with the Lok Sabha, that the lower house of the Indian Parliament. So this is the general rationale behind it, and this is what I wanted to communicate with my students while describing the importance of the Rajya Sabha. Similarly, coming to the scientific rationale behind it, you know that as the Chaiwala had two, you can say one glass and the other pitcher. So two pots, we can say that he is having two pots. Two pots means there will be more surface area and there will be more evaporation. Because of evaporation, the tea got cooled. So these are the two rationales behind this story I told you. And coming to my topic, next slide. So today I am going to deliver my talk on development of new and innovative courses and curricula being organized by International Journal of Microbial Science in collaboration with Sushila, Shankarrao, Gadhave, Mahavidyalaya, Khandala district, Satara. I am thankful to the IJMS for giving me this beautiful platform to deliver my talk. I am also thankful to my dear valued participants for attending this 1 week faculty development program on statistical analysis in academics and research. Now let me start my topic, delivering talk on my topic. Next. So Prime Minister Narendra Modi has rightly said that education has an important role to play in making India self-reliant. It is true, but for that, we have to bring innovation into the classroom. That is the need of the hour. Next. Now, what is innovation? Coming to innovation, what is innovation? We know very well that there is no dearth of technology in India or in the globe. So when you combine the creativity with technology, then it becomes innovation. So the need of the hour is to bring innovation into the classroom, to make the students attentive and to keep them more engaged. Next. And the Labour President, who is popularly known as Dr. APJ Abdul Kalam, rightly said, learning gives creativity, creativity leads to thinking, thinking provides knowledge, knowledge makes great. That's why creativity plus technology, we have to mix both creativity and technology to get new innovative ideas in our classrooms. Next. Yes, there are many innovative ideas that we are adopting in our higher education system, starting from 3D printing up to ILMS, that is improved learning management system. In between, there is free tuition, virtual reality, video streaming, artificial intelligence, learner faculty forms, and competence-based learning, all these things. So, but I have to cover all these things within very short time. So without wasting time, let us move on to the topic. Next. We know very well that this COVID-19 pandemic has changed the dynamics of the classroom, be it primary education, secondary education, or higher education. Now, the chalk and talk has been replaced with click and talk. That means we have switched over to, you can say, that virtual meeting space. So this COVID pandemic has in fact brought a dramatic change in this education scenario, and it has brought many changes in the pedagogy of education that we are using since long. Next. Next, man. Yes. So what is the agenda of my topic today? So it is storytelling, virtual classroom, artificial intelligence, video streaming, digital textbooks, competence-based learning or competence-based education, CBE or CBL, 3D printing, and the last one is ILMS, which is called Improved Learning Management System. Okay. So as I have to cover at least 7 to 8 topics. So let us move on to storytelling. Next. So classroom storytelling. So it is in fact a perpetual teaching and learning strategy to keep the students attentive and engaged in the classroom, be it an offline class or a virtual classroom. Storytelling works, it works like anything. Because if you go, a teacher, if you go to the class and direct starts teaching the topic, then he or she may not show any interest in that topic. So the need of the hour for a teacher is to tell a story before teaching the people. We start the teaching methods. So second is provide insight to academic and professional interest. By telling story to the students or the learners, it will not only create interest among the minds of the students or the learners, it will provide insight to them. And it will be a collaborative discussion. The students will participate or the learners will participate, and so that the session or the class will be also interactive. And third, it connects emotionally with the students, because everybody, particularly the young students, they like to listen stories. So even you and me like listening stories. So the stories connects emotionally with the students. And what is important is that if you share your personal experience, as I did minutes ago, or if you share individual stories with the learners and students, definitely it will matter a lot. And the storytelling increase the collaboration, and also it gives spirit to the discussion, so that whatever you teach in the classroom, they will keep it in their mind. Next. And next slide. Okay. Now coming to the second topic, it is virtual classroom, how it works. No, ma'am, the previous slide. Yeah, right. So virtual classroom, how it works. Now we know very well that this COVID-19 pandemic has changed the education scenario all across the globe. Now we have been shifted from a traditional classroom to a virtual meeting space. And it is a new concept to both the teacher and the students. My voice is okay?
Yes, sir. You can continue.
Okay, fine, thank you. And now coming to the second one, that set rules at the beginning. As I told you just now, that both the teacher and students are new to this concept of virtual classroom before this COVID-19 pandemic. What a teacher, sorry, that let me explain the first one. Okay, left, that is prepare in advance. What a teacher has to do, that because I told you that both the teacher students are new to this virtual classroom. So he has to prepare the students in advance. Then he has to supply the necessary resources required for the virtual class. That means he has to prepare the students in advance. And the teacher also himself needs to be prepared in advance. For example, before going to conduct a virtual classroom, what he has to do, that he has to check whether his screen is okay or not, whether his mic is okay or not, what about the sound system, what fonts and template design is going to use, during the virtual classrooms, so that he has to take care of all these things in advance. Coming to the second one, that set rules at the beginning. And what is the rule for the students to attend a virtual classroom? That he has to explain to the students much in advance. And third one is create reward system, because the students are new to this virtual classroom. The teacher has to explain that the teacher has to give many feedbacks to the student, positive feedbacks. That means the motto of the teacher, the very objective of the teacher is to how to engage, how to keep the students attentive throughout the class. And that's why he has to do the needful. He has to give some feedbacks, positive feedbacks to the students, not negative feedbacks. And what is more important is ask more questions. The teacher should ask more questions to the students, because by asking questions to the students, the teacher can keep the students not only engaged but also attentive. So he should ask at least 5 W, that where, when, who, where, and last, that is H, the how it happened. So 5 W, he should ask questions based on 5 W and 1 H. And next is he has to keep the class short and the lesson scripts. So because nobody, particularly the younger generation, they don't like to sit before the screen for long hours. So for that, what you have to do, you have to make the class short. And the lesson scripts, they never likes the lengthy lecture, a lengthy lesson. For that, what you have to do, you have to make the lesson short. And next is do progress checking. And to check their progress, what you have to do, you have to conduct the examinations like assignments, quizzes, all these things, just to jog their memories, to refresh their memories. And next is building student teacher bond. This one is very important. Unless you build a bunch with the students, you cannot win their hearts. So as it is a virtual class, then your responsibility is more, that how to build a strong bond, a strong relationship with the student. Next is use flipped classroom. As we know very well that if you use a flipped classroom, talks about the blended classroom, where you use both the online and offline method. So we have to use both the offline and online methodology at the benefit of the student. Next. Yes, coming to this artificial intelligence, AI, on which I will speak more, I will give more emphasis on this. This age is, in fact, may call it the age of artificial intelligence or AI. You see, you and me, everywhere we comes in contact with artificial intelligence. The artificial intelligence in our pocket. Let us talk about the smartphone we are having in our pocket. Why it is called smart? Because of this artificial intelligence, it is called smart. So there are countless applications of artificial intelligence in our everyday life that we cannot count. But unfortunately, we do not know that we are using this AI. Now, what is AI? Let me explain you what is AI. So it is the branch of computer science that deals with building intelligent machines that can think and respond like humans. So it is the branch of the computer science with the help of which we make the machines, we feed them data, which we may call algorithms. So algorithms are nothing but certain sets of rules which we use for solving various problems. So while dealing with the artificial intelligence, I can explain you that This is the branch of computer science which deals with building intelligent machines that can think and respond like humans. Once we feed the data, then ours is over. Now, the machine behaves like a human being. And you see this man who is in the middle of my screen. So John McCarthy, the American computer scientist and mathematician, John McCarthy, who is widely recognized as the father of artificial intelligence, was coined this term AI in 1956. Okay. Ma'am, next slide. So these are the 6 different branches of artificial intelligence, which have their applications in our everyday life. Let us start with machine learning. What is machine learning? First is machine learning, second is neural network, then your robotics, expert system, natural language processing, and fuzzy logic. Let us discuss one after the other. Next. Yes. So machine learning, as the name suggests, that machine learning, that we learn from the machine. So it is a subset of that AI, or you can say that a sub branch of that artificial intelligence that trains a machine how to learn and requires. In fact, it is the human being who trains the machine, that how the machine should learn, how the machine should respond to the stimuli, and in what way the machine should respond. Second is data preparation capabilities, that once we feed the data into the machine or into the computer system, then we have to ensure that the machine is having enough potency or capability. Second is algorithm. So what is the algorithm? Just now I told you that algorithms are nothing, but it is a set of principles which you use to solve various problems. So in fact, this whole AI or artificial intelligence, including this machine learning, which is the subset of AI, is based on algorithm. And coming to automation and iterative process, that why we called is a automation, because once we feed the data, once we provide the data into the machine, now the machine starts responding automatically without human intervention. Okay. Next is scalability. It's not so much important, and ansible modeling. This is also not so much important. And coming to the applications, that is virtual personal assistants. You know that coming to the machine learning, virtual assistants, that is, you can say that Google Assistant, for example, traffic alerts, Google Translator. It also helps in fraud detection. We use this application for conducting online video streaming and self-driving cars. This is all about the machine learning. And we should feel the difference between the computer science and the machine learning. Next. So next topic is neural network, or called NN. What is neural network? Like our brain, you know that human brain is made up of so many neurons. It is a network of neurons. Similarly, like a human brain, this neural network, which we find in the computers or in the machines, the phenomena is same. So they perform the task like that of human brain. So the neural network performs the same function like that of the neurons of human brain. So coming to the definition, what it is, yes, it is either a system, software or hardware that works similar to the tasks performed by the neurons of human brain. I told you just now. And once trained, they learn on their own. So once you train them, then no need of training them again. They work on their own. And it is a set of algorithms, which I told you just now, that it is also a set of algorithms. This NN concept is based on algorithm. Next, they do this using a process that mimics the way our brain operates. The way the human beings mimics, the neural networks follows that human mimics. They also does the mimic the same way as the brain, our brain does. Now coming to the application, what are the various applications of neural network or NN? Yes, so it helps in speech detection or face detection, image recognition, all these things. You know that while unlocking our mobile phone or smartphone, we use that face ID. And it also helpful in banking, like detecting various banking frauds. It is also useful and applicable to various medical uses and also in defense system. You can see from my screen that how this artificial neural network architecture resembles with the brain neuron structure of human beings. Both of them almost resembles with each other. Next. Yes. Now coming to the robotics, this is another branch of artificial intelligence. So what is robotics? So it is the intersection of science, that is engineering and technology, that produces machines. That means, in other words, we can say the branch of the computer science or the AI, with the help of which we design and make robots, make certain machines which behaves like human beings or humanoids are called robots. And you will be happy to know that India ranks third in implementing robotics automation. 20 DAX robots are already being used by Indian Army. Various universities, colleges offering UG and post-graduation courses in robotics. Even you can see on my screen that on my left side, that is how the DRDO's Lakshya, which is being used by Army to detect the ID. And Mitra in the middle, Mitra was actually launched by Prime Minister Modi in Bengaluru in 2018, and IRA, that is this robot is found in the HDFC Bank in Mumbai. Visit Mumbai, particularly the HDFC Bank, you can see the IRA. And coming to this Mitra, if you are fond of moving, if you are fond of watching movies, you can see in the theaters of Bengaluru, where this Mitra is chatting with the visitors, with the people. Next. Ma'am, next. Ma'am, next slide, please. Ma'am, are you getting my voice?
Yes, sir.
Okay,
I
next
think
slide.
They'll change. Yeah.
So coming to next, that is export system. As the name indicates, export system. So this is also a subset of, or you can say the branch of artificial intelligence, where a computer system emulates the decision-making ability of human expert. So here what we do, a human expert pass the data, or you can say algorithms, to an engineer. Now the engineer feeds the data to the system. So once the data is fed to the system provided by the expert, now the system starts operating. The system works in such a way that you can imagine, because there is no need of moving to or going to a doctor for an advice. If you are suffering from any disease, no need of going to a doctor. Better you should go to an expert system, which is a actually in fact, which is a computer, which is a machine, but he can give you advice. Whatever question you ask, you will get the necessary answers, or even you can get some suggestions, advice from the expert system, because it solves complex problems also. And they use a knowledge base and inference engine. You can see on my screen that how a human expert provides the necessary information, data, algorithm, all these things pass to one knowledge engineer, and the knowledge engineer in turn feeds the same to a machine. Second is human experts are perishable, but an expert system is permanent. And it decreases the cost of consulting an expert. I told you just now, if you are suffering from a disease, no need of going to a doctor. If you want the advice of a software engineer, no need of going to an engineer. Rather, you can approach that expert system or a machine who will, from where you will get the suggestions you require. And coming to the application side, yes, this expert system is used in disease diagnosis. Our health experts, the doctors, they use this technology, this AI technology in diagnosing various diseases. And fault finding. In various automobiles, we encounter various problems. So to find the various faults, we use this expert system. Next. And what is fuzzy logic? It is very interesting. So sometimes actually what happens in certain situation, we are not clear whether a concept is absolutely true or a hypothesis is absolutely true or false. Fuzzy means unclear logic. It is a technique that represents and modifies uncertain information by measuring the degree to which the hypothesis is correct. So, as I told you, that fuzzy means unclear, the logic which is not clear. So, what is the generalization of the standard logic? That it exhibits a degree of truth between 0.0 to 1.0. So, if the hypothesis is true, we can say 1.0. If the hypothesis or the concept is false, then we can say 0.0. But in fuzzy logic, there is an intermediate value which is partially true and partially false. And where you will get the application of this logic or unclear logic? Yes. So this is applicable in microwave ovens, air conditioners, washing machines, and refrigerators. Okay. Next slide. And yes, this is very important subset of, or you can say the branch of natural language processing, NLP. Yes, it is a field of AI in which the computers analyze, understand, and derive the meaning from the human language. That means it is a conversion between the machine and the human being. Remember here one thing, we have fed data We have provided the data to the machines. With providing data, our job is over. Now you can talk to the system. You can talk with the computer, and the computer will understand your language and will behave accordingly. And here also the machines learn on their own and emulate human brains. They are the computational models, essentially the algorithms. So algorithms is the basic of the artificial intelligence. So it relies on machine learning to automatically learn these rules by analyzing a set of examples, because we have feed the algorithms and data to the machines. Now it is the machine, they will make use of those algorithms and data to understand what we want, and they respond in a smart way like the human beings. And coming to the use of NLP, so that is fake news detection. It helps in detecting the fake news and classify the emails. So by reading the heading, or you can say the first line of the mail, the NLP can say whether it is a spam or a junk, that it can identify or classify the emails. And regarding the, that sentiment analysis and personal voice assistants, that Google has, that Google Assistant, and they have also developed that Google Duplex, that is the update of Google search engine. And that Microsoft has Cortana. So these are the various voice assistants where this is the application of NLP. Okay, next. Yes, video streaming. So this video streaming, or you can say live stream of a class, how the video streaming is done and why it is required. Yes, live streaming is a significant tool to stream the classroom session to the students. And during the video streaming, as we know that factors like you see live pools, question answer session, live chats, and many more things takes place, which gives or makes the student attentive and keeps them engaged. And what are the various platforms required for the video streaming? There are so many platforms that we can use for video streaming, and the important platforms are that Facebook Live, Skype, YouTube live streaming, even our FDP is also, it is live streaming. Okay, so how to live stream a class? For that, you have to collect the proper requirements required for streaming, and you have to charge an online video education platform. You have to check your internet connection and prepare your content and start your live stream class. Nowadays, during this COVID-19 pandemic, we cannot deny this video streaming has become more important to teach the students. Next. Yes, digital textbooks. A digital textbook is an e-book intended to serve as the text for a class. So as the name indicates, digital textbooks, or you can say e-books, where you can read it in your mobile, you can read it in your laptop, or you can read it in your desktop. So it is time saving. They may serve as the text for a traditional face-to-face class. You know that MOOCs, Massive Open Online Courses, MOOCs, which has been prepared by the MHRD. Here you can get, you see some thousands or lakhs of e-books. There are many potential advantages to digital textbooks in place of the traditional books. Next. Ma'am, next slide. Ma'am, next slide, please.
Yes, sir.
Ma'am, the screen is not visible.
Yeah.
Ma'am, next slide, next. Next, ma'am.
Is this correct,
Yes.
sir?
Yeah, correct, correct. So next is moving to next is 3D printing. So 3D printing is a recent revolution in higher education. So whether it is a necessity in higher education or not. So you see that 3D printing is a process for making a physical object from a three-dimensional digital model. And it brings a digital object into physical form by adding layer by layer of the material. And nowadays, the importance of the inclusion of 3D printing in professional degrees and in schools and also in college already a fact. Next one. Now, where we can use this 3D printing technology. In fact, the 3D printers have actually been around for 25 years. But barriers like cost are breaking down over the years. So they are now very affordable and easy to use. And in fact, 3D printing has got the attention of the educators across the globe. Using 3D printers in the classroom, you can, even the biology students can study cross-section of the hearts or other organs. And the history classes can print artificial for closure examination. Architectural students can print new or existing designs. Even design and engineering students can make prototypes of their creations. And even the engineering students can print modified car or robot part. Next one. Yes. So next stage, that is ILMS. So what is ILMS? It is a software application that has made inroads into the classroom. Here, let me tell you one thing, that it is student-centric, that it has undergone a shift from a being teacher-centric to the student-centric. And here, all the things in one platform, you will find all those things required for the teacher and the students. It provides tons of online courses. It reduced the learning cost and the time of travel and space arrangements. And what are the various examples? So examples are the important examples of the ILMS, improved learning management systems, are the Google Classroom and the Moodle. Next. So as I told you at the beginning, at the outset, that this COVID-19 pandemic has changed the dynamics of the classroom, and we have changed from the traditional classroom to the virtual meetings space. But the problem with our teachers is that they are new to this virtual meeting space, and that's why they have to, what they have to do, they have unlearn the process, the teaching method. Because what is important for a teacher is learning, unlearning, and relearning. But the problem with us is that we are surrounded by the myths which does not allow us to open our eyes to the real world. So I would like to conclude with a quote uh, from the great American writer Alvin Toffler, who has said that the illiterate of the future are not those who cannot read or write, but those who cannot learn, unlearn, and relearn. With this, I thank you to the organizers, IJMS, and the participants, and I hope the participants have learned something from this talk. So thank you.
Thank you, sir. Thanks a lot. Beginning the question answer session with the famous quote of Anthony Robbins: successful people ask better questions and as a result get better answers. I, Dr. Sheetal Banerjee, welcome you all in the question answer session and request all the participants to post their queries in the chat box. All the participants are requested to post their queries in the chat box. One of the question which I got over here is that, sir, how we can use these pedagogies in while teaching the practicals in science subjects?
Yes. Uh, uh, as I told you just a couple of minutes ago, that what is pedagogy? Pedagogy is nothing but the science of teaching is called pedagogy. And this COVID-19 pandemic has changed the entire scenario. And we are new to this virtual meeting space. And you see that because of the COVID-19 pandemic, all the educational institutions have been closed. And that's why the school, college, and universities, they have adopted this virtual method, or you can say they are conducting online classes. But no pedagogy, no specific pedagogy. I cannot suggest a specific pedagogy to teach the students. But what the teacher has to take care that how he can keep the students attentive and engaged in the classroom. That is the job of the teacher. And for that, he has which pedagogy he has to adopt, which methodology he has to adopt. It is up to the teacher.
Okay, thank
I
you,
hope
sir.
it is fine.
Yes, sir. Thank you, sir. If any of the questions are from the participants, is most welcome. You can put in the chat box so that I can pick it up. It seems that the participants are most satisfied. Uh, thank you, sir. Thanks a lot. On behalf of organizing committee, I thank you for this insightful session. I hope that your expertise will definitely help the participants to enhance their knowledge. Thank you. Thanks a lot.
Thank you, ma'am. Thanks a lot.
Over to you, Naz, ma'am.
Yeah, thank you, ma'am. And indeed, the session was really good, and it gave us a lot of insight on how next learning system will be. And definitely, all teachers are always learners before they are teachers. And that's why I can see this FDP is going to be a successful one, because all the participants who have come here have come with the thought of learning something new. And always there is a new beginning. And so, which we couldn't do in 5 to 10 years, this COVID pandemic has done in 5 months or 6 months, that we have all gathered together on this virtual platform from all over the parts of India and international as well to come together to a learning experience. With this thought of ours, we would be ending this second session for the day today, hoping to meet for tomorrow's session. And tomorrow's session, we have two speakers. For session one, we have on use of statistical package for the social sciences, that is SPSS Statistics version 23.0 to plot interaction effects by Dr. Kishori Kashyap, ma'am. And next we have Institutional Governance and Policy document by Dr. Shubha, HOD and Associate Professor of Botany from Government First Grade College, Vijayanagara, Bengaluru. So, coming to an end for this session, before we wind up, I would like to highlight some points for the participants. Participants are hereby informed that the feedback link and the MCQ exams link will be given on the 8th November 2020 at 7 o'clock sharp in the YouTube link description. So, or in the website, that is www.theijms.com. And there is one more news here for all the participants who have missed out to register themselves can do the registration, because we have kept the registration link open as we got many queries about registrations to be fulfilled, because many of people are wanting to register themselves for this FDP. And those who are wanting to register, the registration is free. And those who want the certificate can pay a minimal amount of these charges for the certificates and you can get it. And definitely this FDP is going to bring a lot of changes in your API score as well. And the next thing is, I wish all the participants better learning and useful learning. And with this thought, we would be coming to an end to this session of ours, hopefully with all of you. And definitely, I would like to tell all of you to stay home, stay safe, because these times are testing times for all of us. So for this, signing off here, Alianas, and with my team of IJMS, in coordination with Sushila, Shankarrao, Gadhave Mahavidyalaya, taking care of all of you in your fields. Thank you. Have a nice evening. I can't say. Actually, we are towards night. So good night and sweet dreams, all of you. Meet you tomorrow sharp at 7.